one of the things I personally learned about the article and about RNA interference in general is about like the cellular mechanisms that occur. So, for example, I don't know if you guys were able to look at figure one, but figure one was very interesting. It talked about how small interfering RNAs they enter the cell, and then there's dis no, there's dicer, and then there's risk, and then everything ends up working together in order to silence the mRNA that they're trying to silence. So I found that very interesting.
Yeah, well, for me, I guess it was like mostly the topic of siRNA and shRNA and how they're like kind of they share like the molecules, but they kind of, are different in the outcomes or let's say the effects. So that the shRNA, I guess, is long term and the siRNA is for like acute diseases versus the shRNA being for longer, chronic diseases, let's say. And that, shRNA has like, you know, doesn't have that much of effect and but siRNA, has chances of cytotoxicity and inflammation. So I guess it's kind of interesting to see how these two, while kind of similar, they have like, different effects. I guess that's like kind of something that interested me about it.
I agree. Yeah, I was absolutely shocked whenever the article mentioned how like shRNA can like last up to 6 years, I think it said, which was really surprising to me. I didn't know therapy could last that long, but I completely agree with you. That was very interesting.
Yeah, and siRNA would like 46 hours, I guess. So yeah, it was like really contrasting.
Mhm. Yeah, I love how you pointed out the differences between siRNA and shRNA. I know many people get them confused because they sound so similar, but I think you did a really good job explaining the differences. It's very,
Oh, thanks. Well, there are a lot of types for RNAs, obviously, so yeah, it should be confusing.
yes, very complex.
Yeah, that was interesting to me as well, especially because like whenever I was in the lab or whatever, we were always talking about how delicate the RNA was, but then the fact that it can last up to 6 years being that delicate was really interesting.
Mhm. Yes, definitely.
I also found table one really interesting with the ncRNA and the different classes and the characteristics and everything. Because I just found it really interesting how like a difference in one nucleotide is making such a difference in the characteristics and whatever.
I agree. Yeah, like for example, I'm like Avani and I, we both are taking a class called genetics in our school, and the teacher like mentioned so many times how just like one difference in the nucleotide can like change everything. Like it can cause sickle cell disease and everything. It's just absolutely fascinating and kind of scary, but yeah, I agree with you. It's definitely very interesting. Emma, what about you? What did you find interesting?
Found it interesting how 'cause I'd heard about this the RNA therapy before, but I wasn't entirely sure how it worked. So I thought it was interesting hearing about the gene silencing and how it works to turn off the genes. I also thought it was interesting. It talked about synthetic RNA or like man-made RNA, which I thought was cool. I didn't know that that was a thing that we could create RNA. That sounds interesting.
Yeah, I wonder how they make it. I wonder like if pharmacists make it or like biochemists. It's definitely very interesting to see how they would make it and synthesize it and then test it to see if it works.
Right? Like how hard is it to make? I'm sure it's pretty hard.
Yeah, I feel like in genetics, especially with like, gene silencing and epigenetics, I feel like there are so many things that can go wrong, like offset targets and stuff, offset target effects and everything. And I find it really remarkable how scientists have understood these really molecular microscopic mechanisms and then have been able to use that to our advantage. I find that very interesting. Yeah, and RNA interference just seems like such an innovative treatment, and I really hope it can help cure many diseases in the future, especially because it has promising effects, but I feel like it might not be entirely understood as of today. So I really hope more research comes among that. Mhm.
That was one thing I found interesting at the end of the article. Because the whole article, it seemed to have a very positive spin on it, but then at the end it said it systematic delivery to tumor targets other than the liver has resulted in many problems. So I'm curious what the cons of it are.
I'm where exactly is that? I'm trying to find the sentence.
It was on page 318 and the conclusion, like the bottom of the page for the conclusion.
Here, let me see if I can share the screen just so we all can see it. I don't know why it's not letting me share my screen, honestly, but I do see where you're seeing that. And I agree, like I didn't really know. I've heard of RNA therapy and I've heard of all of the positives and all of the potentials of RNA therapy, but I really also liked how this, as you said, this article also mentioned possible negative effects of it. Um, so were you guys able to also find any other negative effects in addition to, for example, the liver? Or can you guys like think of any possible negative effects that RNA therapy could have in science and in therapeutic interventions?
Well, because RNA is so, it's not like DNA where it is what it is. I mean, to some degree, but it's going to change and whatever. It makes me wonder if, yes, it has a lot of promise in the RNA that we're able to target, but then is the cancer going to figure out how to mutate to avoid the target?
That's actually a really good question because I know cancer cells, they have like such a brain of their own kind of where they can just, as you said, like they can just mutate and they can just evolve kind of in order to bypass the different, interventions that people can give. And yeah, I think that potentially cancer cells could do that. And I also wonder, could RNA therapy potentially cause cancer or maybe exacerbate cancer? Because, for example, if something goes wrong and the, synthetic siRNA or shRNA, if they end up binding to the wrong gene, potentially, that could mutate it and end up causing something really unintended.
Yeah, and another thing kind of down that same road is, you know, if you have a genetic disorder, it's not like you're going to develop one, but I know that some people just are like carriers or just have like the potential or whatever. So this makes me wonder if we turn on or off the wrong gene, are we going to start not causing because the potential is already there, but are we going to start triggering um like uh physical, like are we going to bring out the disease as a disease within genetics if someone has that potential?
That's a really good point because I know in, biology class, we learn how even the smallest thing can cause a cascade of events that makes the human body, for instance, like conducive to various diseases. So I think you're definitely right about that point, and it's very interesting to hear. Yeah, and I'm wondering, I think another con about RNA interference might be the approach to actually delivering the RNA into the human body. I know current research sometimes uses nanoparticles and different types of vectors in order to introduce it, but it might be difficult to actually make sure it's utilized by the cell rather than just excreted by the body. So that might be one of the downfalls as well.
Yeah, I've heard of in gene therapies them using virus vectors, and part of me wonders if like they can do that to get it into the cell, but then there's so many. Yes, it's so promising that we can alter RNA and even DNA to some degree, but then it's the trade-off. What else are we causing? And another part of me thinks like, if we can use this to treat cancer, can we use it to treat other things? Can we use it with genetic diseases? Can we use it with autoimmune diseases? That type of thing.
Hmm. That's a good question. Yeah, I know that this article talked a lot about cancer, but I believe RNA therapy could potentially be used in order to cure other genetic diseases like Huntington's disease and muscular dystrophy. I wonder if, do you guys know if there's something called DNA therapy as well? Or do you guys know how gene therapy works? This is kind of a separate question.
I actually saw an article on this once. I can't remember everything on the top of my head, but if I find the article, I'll send it to you. It was pretty interesting. But I know that they I don't know how, but they somehow find a way to alter the one specific gene they want, and then usually they use a virus vector and then use it do and then get it into the body through an infusion.
Oh, interesting. Yeah, because I was wondering, because I know gene therapy, it can be used to cure different genetic diseases. So I wonder if RNA therapy can be maybe used alongside gene therapy in order to kind of, optimize it and kind of like offset the negative effects that we just discussed about RNA therapy. I don't know if it would be very expensive to do that, and I'm not really sure about how that would work. But what do you guys think? Do you guys think that scientists might use gene therapy and RNA therapy alongside each other, or would they prioritize one over the other?
I think that that would be really cool. There was an article I just saw from the Children's Hospital, and they just had their first patient recently that they did gene therapy for muscular dystrophy. And I was actually talking to a nurse about it too. And I think that scientifically and medically, it could be very promising to do the two together. But I don't know how realistic that would be, especially as far as insurance companies go, because for that one um, gene altering infusion, it was $6 million for the insurance company. So
Oh, wow. Yeah, I feel like a lot of scientists, like we, learn all of these different things, and then we're like, we can do this and we can do that. And then we sometimes forget about insurance and monetary stuff. Like I always do that. I always am like, why not just use CRISPR and solve, cancer and stuff? And then my teacher has to remind me that money is definitely a problem. But yeah, so what did you guys think of the article overall? Was it informative and were you guys able to, why don't we all share one thing we learned? Do you think that would be good if maybe we can like discuss some key aspects of what the article talked about and what did we learn about that? Yeah, I mean, we can take, a few seconds to just look over our notes and then whenever you guys are ready to, you know, reconvene, we can definitely discuss. Oh, Hadi, I think you're muted. I can't hear you. Yeah, maybe. Want to start discussing the points of the article and what you guys learned about it? Or do you guys want some more time? Uh, are you guys able to hear us? I don't think. You can? Okay, good. My internet's being so weird today. I really don't know what's happening. Um,
I guess it's working now.
yes. Okay,
Yes.
now we can hear
Oh,
you.
okay, great. Okay.
Okay, I
Yeah,
think
so
you were mentioning something before we weren't able to hear you.
yeah, I was just, commenting on the last thing that you've been talking about, which is I think like the practicality of the method, it could just, come at a later time until we could fully grasp the process and until they want to implement it, even if let's say, you will put effort and then eventually you will do nothing in the real world, despite that, it can still be, useful and maybe it's just that at a later time we would figure out a better way to implement it, regarding resources, regarding yeah, like what resources people or hospitals have access to, and not, just the exclusive, medical service that some people and, the expense of, let's say, medical service that some people would get. So apart from its practicality, I guess we should study first how it really, its alterations, it if it would, just exert any negative effects on the human body, and if it would really affect the genes, apart from just treating the tumors and stuff. So I guess that could be, main focus if it's going to be really taken seriously.
Yeah, I wonder if they're doing clinical studies in order to, um, in order to see all of these, like the negative effects like you're talking about. But that's definitely something we would have to do some research on outside of this in order to see, but that's definitely good points.
Well, I guess we don't have simulations for the human body to be that accurate, so I guess we can do it, at least, in the foreseeable future, like like the next several years, but maybe later or as it advances more, the technology and stuff that that we depend on to implement all of these studies, and because it's just theoretical at some point, and you need something to because in reality even the most, let's say, ideal conditions that on which we would base off our studies and the ideal human body and everything, it would still have there will still be interferences with the real image, with the real individualized image of every body that would benefit or would undergo the treatment.
That's a very good point, especially because I know many treatments have different side effects, which make it very difficult to actually bring it to the overall population. And I feel like personalized treatments are also an interesting thing that you mentioned, as every body is different and it's important to take that into consideration. I also feel like the article mentioned various clinical drugs that are using this concept. I'm not entirely sure about that, but let me check because that would be very interesting if this is actually being used in modern day society.
The article did mention three specific drugs that said were in phase 1 studies, but I read a book on it was on the COVID vaccine, but the people who made it, the company, they specialized in cancer RNA research, and they briefly talked about that in the book. And they made it sound like that company had done some studies with people, and they didn't touch much on that. But I think it's interesting to think about like where it's at now. It would also be interesting to think like if it fixes the problem, it might fix the problem for the person who gets the therapy treatment. But I'm curious how it would affect like future generations if it wouldn't show up until future generations' negative side effects.
Oh, so I guess so. Are you wondering, like, let's say that there is a mother, for example, and she had received treatment of RNA therapy. So is your question like, how would her kids like? would they inherit that kind of like the effects of RNA therapy, or would they have the gene?
Yeah, that's like how it would affect because it an inherited trait and how that would work, I guess.
Such a good question.
Yes.
Because I know in class we learned about epigenetics and how like epigenetic changes are passed down from parents to offspring. But that's a really interesting question how if you synthetically change the expression of genes, would that end up being passed down, or would the natural set of genes be passed down? Do you have a comment about this, Lily or Hadi? Do you have any ideas?
Well, part of me wonders if it wouldn't be passed down because it's not in the DNA itself. But then if we are changing the gene, then it's kind of what would it be passed down? And then another thing is if a mutation in the gene is causing problems in the first place and that's why we fix it, is it actually going to be beneficial for the future generations? So I don't know, the breast cancer gene, I forget what it's called. But I don't know if that is a gene that is passed down or not, but I think it is. So like if there was something that could alter that gene, well then would it be good if it was altered in the offspring? That's kind of. And
Yeah.
then if it is altered in the offspring, how many generations does that go?
Are you talking about the BRCA gene? The
I
BRCA?
think so, yeah.
Yeah, I've heard that that is passed down, and that's a really good question about ethics as well. Is like, should we end up like tampering with those genes and like all of that stuff? That's definitely a big question I think scientists are asking right now. But yeah, it's definitely a very interesting question, Emma. That was a really good question. I think it got all of us to think about, how would generations, like future generations, be impacted by RNA therapy? And I wonder, I know that like shRNA, for example, it ends up, silencing the gene for a long time. I wonder because shRNA and siRNA, they don't like they silence the gene by targeting the mRNA, but the gene is technically still on. I wonder if it would not be passed down because if the kid ends up inheriting the gene, won't it still be on? And maybe they would have to also like undergo RNA therapy or an infusion in order to end up silencing that gene. Does that make sense? I'm
That
kind
does make sense because I forgot that it was still technically on.
of, yeah, very interesting. I mean, I don't know. Maybe scientists will figure out a way in which like I don't know. I don't know if this is like too sci-fi or something, but maybe scientists will figure out a way that one day, like the mother or father can undergo RNA therapy, and then that could, end up changing the genetics for, the kids for a more positive note. But I think that's very sci-fi. I think we have a long way to go before that would happen.
It is an
Um,
interesting thought, though. Like, if they do in utero testing on a baby and that baby is going to have a genetic disorder, I wonder if the parents can undergo, like, the mother can undergo treatment before the baby is born and it will have a positive effect on the baby. I don't know.
Wow, these are such good questions, guys. This is such a good discussion. And Holly,
well, okay. Oh, sorry.
were you saying something? Sorry,
Oh, yeah. I mean, no, no. I mean, excuse me. If you want to say something, go ahead.
oh no, no, you can go.
Okay, thanks. So yeah, I suppose that, today there are some main concerns about, let's say the surroundings, or let's say the climate generally, and the lifestyle, let's say in the upcoming, decades for, the future generations, and how they are going to be affected by, environmental pollution if we are causing any. I guess I saw something about this. There was like research, there were studies, there were polls, there was, public, a case of public opinion about that topic of like things related to pollution and how it would affect, future generations. So apart from let's say the outer side, let's say for the inner side, what's happening inside the body. I mean, in the scope of the biological build, I guess, yeah, studying the transmission or the potential of these genes, let's say altered genes being transmitted to the offspring, like it is crucial, but it also is still kind of personalized. It depends on like if we're discussing, making the mother, the focal point of it. So it also depends on the father. And I guess it should also depend on, further interventions, with the genetic build of the larger family, not just, the couple. So this should also be taken and, for it to, yield the best results and not cause any harm to the offspring. But I still, yeah, like we've come a long way, but we still got a lot to do. And it's not that ceremonial. Like we can't just celebrate what we did without thinking of the consequences because, we would just study what we're studying in the scope of today and maybe would not think much about tomorrow. But yeah, I guess we should first think about, how we can implement it. Um, you know, currently, and then we would think about its consequences on the offspring, the people who are gonna come after us, the generations, and then we can study how we can implement it, in terms of the real world and the medical resources that we have.
Yeah, these are all really good ideas, and it's definitely a really good point in order to take into account also like the general family history and family genetics instead of just the couple. I forgot that, it's not just the mom that ends up passing the genetics, but it's also, the father and it's also like the grandma, like indirectly, it's a really long chain. So that's a really good point. And then also, I wanted to let you guys know this meeting is going to end in 10 minutes because of Zoom's time restriction. But I wanted to ask you guys, do you want to dive into the figures and start discussing what all the figures are talking about? Are we ready for that? Yeah.
I guess it's not a wrong idea.
Yes. Okay, sounds good. So I am going to try to share, but while that, do any of you want to try explaining figure one? Yeah, I can give you guys like a few seconds to look at it while I try figuring out how to share my screen. But okay, I was finally able to figure out how to share my screen. So here we go. Are you guys able to see the screen?
Yes.
Okay, perfect. Perfect. So I'm going to scroll down to figure number one. Um, okay, here we go. So here's figure number one. And so do any of you want to explain it to us or try to explain it, or we all can also discuss it together. So what do you guys think about this diagram? I know there's a lot going on.
I think it's mainly just showing how the risk is cleaving the RNA. I think that's kind of the main point of this diagram.
Yes, I agree. And trying to think. So, yeah, I found it really interesting how the diagram shows two different ways that we can introduce siRNA into the cell. We can use a vector, and we can also use a hairpin siRNA. And yeah, so how do you guys think that it's introduced to the cell? Like, if you guys understand my question, do you guys have any ideas of how the cell absorbs it or engulfs the vector or the hairpin RNA? Yeah, I think,
Well, sorry, go ahead.
sorry, go ahead.
For the vector, I feel like it kind of depends what type of vector you're using with how it penetrates the cell, too.
Like, what different types of vectors are there?
So I know there's viral and bacterial vectors, and then I think there's a few more as well.
Oh, okay. Yeah, I'm not sure if nanoparticles is considered a vector, but that might also be a possibility of introducing it. Hmm. Yeah, and I know you were mentioning viral, like viral vectors as well. I wonder if viruses like bacteriophages or other viruses, if they like if we can like in the lab, if we can introduce the vector into the virus and then the virus ultimately is going to, inject the vector into the cell. I wonder if that's also a way that it can be introduced into the cell. Yeah, I feel like I've heard that they use the viral vector concept to help treat cystic fibrosis. I definitely need to research more about that, but it seems pretty awesome.
I think they use it for sickle cell as well.
Oh, that's very interesting.
And I know they use it for muscular dystrophy in some cases.
Hmm, interesting. So, Emma and Hadi, what do you guys think about this figure?
Okay. Sorry, I don't know if I'm missing anything, but what is the difference between the synthetic siRNA and the hairpin siRNA? Like, I mean, the moment it passed from this you know, I'm not remembering the word, like, it passed this wall.
The cell membrane?
What's the difference?
So, are you asking like what's the difference between like the vector and the synthetic siRNA?
Oh, no, I think I
Yeah.
see what he's saying. How when it's outside the cell wall, they call it synthetic, but as soon as it goes inside the cell wall, they call it hairpin.
Oh,
Okay, wait. So this is happening. So this is happening in the hull of the cell.
yes, this is a cell
It was
membrane.
all inside the nucleus.
Oh, I think this is the nucleus. It has,
Yeah, yeah.
yeah.
Yeah, I can see, but I thought I don't know. I just thought that. But yeah, okay, it's cool that it's happening inside the and I guess that it's saying that it needs ATP, so yeah, it kind of makes sense. But the drawing doesn't, make it clear a bit.
That's a very interesting question. Yeah, there's definitely a lot of things going on in this diagram. I definitely understand why it might be a little confusing.
It's pretty self-explanatory, I guess, but it probably needs a bit of, background clarification to it. But as I told you, Bhumika, because I haven't read the article pretty well, I haven't researched the topic pretty well. That's why I guess I'm not really grasping the diagram or the figure.
Yeah, I mean, we can go over it. So, yeah, and so what I understood from this diagram personally is that the scientists they introduce the siRNA, whether it's in vector form or whether it's, the hairpin form. So they introduce it into the cell, and then with the hairpin siRNA, Dicer ends up making it from double-stranded into single-stranded pieces. And then what happens is that the risk complex, so the risk complex is just a complex of proteins. And so what the risk complex does is it ends up getting the guide strand of the siRNA, and then two things happen here, I believe. And so one of them is that the siRNA and the risk complex, they bind the mRNA, and therefore the ribosome can't attach to like the siRNA mRNA conjugate matrix. And then the second possible pathway is that the risk protein just cleaves the mRNA. That's what I understood. I mean, do you guys have any like ideas about what the diagram might be saying? I
That's what I thought as well. One question I did have though is why it was double-stranded in the first place when I understood that RNA was usually single-stranded. Um, do you know why it's double-stranded in the first place?
I believe that it's double-stranded because naturally the complementary, like some RNAs are so long that whenever they fold together, it's due to like intermolecular attractions, like hydrogen,
Okay.
because of complementary base pairing. That's
Yeah,
my thought process about that.
that makes sense.
And I don't know if this makes a lot of sense, but I guess like that they've written here, siRNA duplex. So it's just this single stranded piece that it's labeled siRNA, but this one is labeled as duplex. And I guess from what we've been learning in school, mRNA is, that's consistent with what I know, about mRNA, which is coming from inside the nucleus. But because this is labeled like duplex and the mRNA is single stranded, so I guess there's nothing confusing about this, but I guess that's right.
I think it takes some time to digest, but yeah, I think that's all good.
Okay, so first I looked up some terms because there were some things here I'd not heard of. So the SH RNA is short hairpin RNA and it silences genes. And then when it says the expression vector, I'm not sure specifically what expression vector they used, but I looked it up, and for shRNA, the most common vector is a lentiviral vector, which is a type of virus and is most commonly, like the one people most commonly know of is HIV. And then I was looking up pre-shRNA, which is double-stranded and is produced by shRNA being processed in a nucleus. And then the P bodies are processing bodies, and they're ribonucleic protein granules in the cytoplasm, and they store RNA. So I think what I understood was just that they were using the expression vector to get the shRNA in and to silence the genes was kind of what I was getting out of this. But I'm curious to see if you have any more information than that, Dominika.
Yeah, I mean, that's a really good explanation. And whenever I was looking at this diagram, it really reminded me as like an extension of the first diagram, kind of. But I feel like this diagram was mainly elaborating on how does shRNA, like, it was taking a step by step from the nucleus point of view and then how it's transported out with the exportin protein into the cytoplasm, and then it ends up, doing its gene silencing mechanism. And one thing I was unable to figure out, so I know you mentioned this about P-bodies, but I was wondering, are P-bodies like the complex whenever ribosomes attach to the mRNA? Or like, can you explain that? Because I wasn't able to figure it out myself.
So I'm not 100% sure, so just keep that in mind. But from when I was looking it up, it seemed like they were kind of, it seems like they were already there, like not necessarily formed by something. Um, because their job is to store RNA, and so even when new RNA is not introduced into the nucleus, it seems as though they even store the regular RNA that's already there without introducing anything new. But that was just my understanding. I'm not 100% sure.
Very interesting. Yeah, and yes, and then another thing I wanted to add, I found it just really cool how they use viruses in order to do this, because I feel like oftentimes viruses have a very negative connotation, but in this case, I feel like viruses, it's really interesting how scientists can use viruses and then inject the shRNA, whether it's in like a plasmid or a vector or something. And so they can just use viruses, inject the shRNA, and then that can do so much good. I found that so interesting because whenever I was growing up, I always thought viruses were really bad. But
Yeah.
but yeah, I think you explained it perfectly. I don't know what else to add.
Okay, so I forget who's doing figure three, but are you ready for us to go on to there?
I believe Hadi was doing figure 3.
Okay, I'll just go on to there and we can discuss it.
Okay, Hadi, you can take it away.
Okay, great. So, yeah. So first thing, as I guess y'all can see, first we have an RNA polymerase II promoter. I don't fully grasp what this actually how it functions, what it does, but yeah. And we have a cleavage-dependent short hairpin RNA and a cleavage-independent shRNA. And I think from what we can see down in the description, like at the central location base 9 to 12, I guess that node, you know, kind of like midway between the bottom and the top, uh, in the cleavage-independent short hairpin RNA figure that we can see. And there's a TRXN terminator. I'm not sure if that means thyrodoxin or um whatever else, like what abbreviation it is, because and I looked it up, I cannot find it. So I don't know if you have any clue what this is. And then we can see that they all, kind of come together. They're grouped and they form kind of like a bifunctional short hairpin RNA, I think. And for the quadruple A notation, below, I don't also know what it really means, but I guess that the TRXN terminator kind of plays a like a brokering role, and then the cleavage independent shRNA, the cleavage dependent one, like they're the main components of it, and the RNA polymerase, it's a promoter, so I guess it's kind of like the base on which you know like they just get connected, and I don't know how to explain it, but yeah. So then, in kind of like, I guess what they mean by dicer and cleavage, RNA-induced silencing complex, is that it kind of gets divided. I don't know what what kind of simulation we'll have here, but obviously they're kind of splitting. But if the drawing is not, you know, making clear, maybe they're not splitting. Maybe this is just how they're still like in the same form, but how each one kind of morphs. And so here we're gonna be switching, I guess, from bifunctional short hairpin RNA to pre-interfering RNA, because this is what we're gonna want to get in the end, something simpler but more functional. And so here we have the cleavage independent shRNA becoming a rapid target mRNA. It's like translation repression, so it kind of translates weakly, although I don't get much of that what that means as an interfering RNA. Then we have the cleavage dependent short hairpin RNA becoming a target mRNA, not a rapid target one, and it degrades over time. So both, I guess, they're similar but different at the same time. So the scheme here is not that clear, but it depicts how bifunctional shRNA kind of morphs into mRNA, which is like both like target mRNA, and they kind of degrade or are repressed over time. So yeah, and the aim is, as described below, is to activate both the RNA induced silencing complex, dependent cleavage, and the dependent cleavage part. So that's it.
Yeah, that seems like a very difficult and complicated diagram, so I think you did a really good job explaining it. And I think,
Well, thanks, but I think it's just like it's unclear. It's not that hard, but it's unclear.
yeah, and I think one of the reasons the diagram may be unclear is because it's talking about bifunctional shRNA, which I know is like a step further from, the typical shRNA mechanisms that we discussed. And so I believe as bifunctional shRNA, it's complicated because it targets multiple pathways, right? Like the risk dependent and risk independent pathways.
Well, whatsoever, I don't have much of background for this, so I guess it's my fault here. But again, I didn't find it much graspable, we can say.
Yeah, I also found this diagram to be very complicated, and it took me a lot of tries to actually try to decipher what's going on. But I feel like, as you said, the main concept from the diagram is that the two different sections of the bifunctional shRNA, they both are used in like multiple pathways for gene silencing, or not gene silencing, but mRNA degradation and translational inhibition. So that's basically what I got from the diagram. I'm not sure if anyone else has another idea about what's going on.
I didn't really understand it either as well, but one thing that's interesting about the bi-shRNA is that it says it's newly designed RNA interference. So that probably plays into why it's harder to understand and you can't find information on it because it's newly designed. And I also wonder if that makes it sound like it's synthetic, and that probably makes it harder to understand since it's not organic.
Yeah,
Okay.
I
If I
oh,
might want
you
to,
can go
oh,
ahead.
sorry.
Oh, you can go ahead. I didn't mean to interrupt you.
Okay, yeah. If I might want to add something, I guess if it were just, if it's made clear what they meant by TRX and Terminator being a factor in the process, so I think this is important. And with the quadruple A base, like in the second like below the first step, I can say what it stands for and what they mean by dicer and cleavage RASC, like how it plays into it, and whether step two and step three or um let's say in regards to the timing, like they're like separate steps. That's what happens. They they kind of get together first, the cleavage independent, the cleavage dependent siRNA, and then they split again, but something has changed. So yeah, I guess if they just clarify what the role of the terminator or inhibitor or whatever it is, and what the RNA polymerase does, and how these will turn out to be interfering RNAs, I guess it would have been really, pretty clear because I guess it's an infinitesimally short process. So I don't think we need much of a background to it because it's not that clear, but it's still kind of simple. So that's the point.
Are we ready to move on to the next diagram, or? Yeah.
Um, so this one I found it's it's a lot easier to understand because it's more words. But um, this figure illustrates how they create the process for making the drugs for the treatments for cancer. And I find this really interesting how they make it personalized to each person. So they have to have a biopsy of the tumor and extract the RNA and the DNA, and it says protein profiles from the tumor. And I believe what they do is um, I guess it's sort of like finding an antidote for a poison, but they're finding the RNA that will um, attack the uh, genes of the cancer tumor. And I think that's all I found out about it. I was just like each word that I didn't know, I was looking it up, so I didn't get very far.
Yeah, I personally also agree with Emma. I found it very interesting how they use the RNA interference science as sort of a precision medicine where they can alter the treatment depending on the individual. And I believe like personalization in medicine allows for mitigating drug resistance as well as accounts for differences in tumor behavior in various individuals. So I found all of that to be very interesting. And I really like the diagram because, as Emma said, it wasn't as complicated as the other diagram, especially figure three. And so I really thought it had good therapeutic efficacy and it was a pretty good diagram, I feel.
One question I have about this diagram is, so it's saying that the tumor can be harvested with the biopsy, and then you can introduce shRNA in order to target those proto-oncogenes or oncogenes, the cancer-causing genes. And so I'm wondering, does this only apply to aggressive cancer tumors, or does it also apply to benign tumors? Like, do you have any idea about that? About which types of tumors it works with, or maybe if there are specific cancers that work better with this than other cancers? Does anyone have any ideas about that?
I do actually have a question about what you just said because I don't know, I was under the assumption, and this might just be completely wrong, but I thought that most benign tumors did not have their own set of genes because they were benign. I thought you could only have, I thought there was only a separate genome kind of genetic makeup if it was cancerous. So do benign have their own, set of
I
genetic?
think that's actually a good question. I was under the impression they might because it's abnormal growth, but I know benign tumors don't cause any harmful effects. I think they're just there. But I'm gonna search it up because that's actually a really good question.
You
Yeah, oh, sorry. Go ahead, Emma.
well, I was just gonna say, Bhumika, you said something about what types of tumors, and it does say in the conclusion, um, systematic delivery to tumor targets other than the liver has resulted in any problem. So I guess it works best on, I guess, liver cancer.
Hmm,
But what you have to say is interesting.
very interesting. I must have missed it, but thank you.
Yeah, and to answer Lily's question to the best of my ability, I was thinking that benign tumors also have mutations like, oncogenes like P53. I was just under the impression that in the metastasis of like systematic cancers, it's mostly because of the human body becoming conducive to that type of cancer spreading and everything. That was what my perspective was, but I think maybe Bumika would have a better answer than that.
That makes sense though.
Yeah, I mean, I'm still hunting for an answer from a reputable source, but from like the initial look I got, it says that benign tumors do have mutations in their DNA when compared to malignant tumors, but benign tumors have less mutations than malignant tumors, and that's why they don't have harmful effects. But I'm gonna need to do more research on that and get like a more definitive answer.
That makes sense. I'm going to research it too, and I will let you know what I find.
Yeah, that would be wonderful. Yeah, we can just email in the chain and then have a discussion like that if we want. Because that's a really good question. I never thought of it that way, but this is why I like Journal Club. It, you know, gives me so many insights I would not normally think of. So,
Okay,
yeah,
so
yeah.
sorry, if I may comment on this. For me, I think this is not much more like informative, um like it's not much of an informative diagram. It's kind of like describing what factored into the process. So there's not much scientific um there's not much scientific definition to this. So this, I guess, would need some, kind of like background knowledge to fully digest and know how it goes, because it's an operational, um like figuratively. This is how we're looking at it through this diagram. But yeah, I guess maybe also referencing resource number 74, reference number 74, I guess would be kind of good, but I didn't do so actually. So that's my take.
Interesting. So does anyone have any other thoughts about the figures or anything else before we wrap up this meeting? Because there are 2 minutes left of this meeting.
I just want to say that I found the article to be very informative and interesting, especially because I feel like popular cancer treatments are chemotherapy, and so this was a shed on new light of RNAi technology.
I agree. It was interesting. I'm also curious with AI, they're like trying to create like health improvements, and with figure four, they were talking about using bioinformatic data, which is this is from the National Human Genome Research Institute. It's used computer technology to collect, store, analyze, and disseminate biological data and information. So I'm curious how, like, if they create like a super intelligence or something, how much quicker that would go, and how this cancer research would work with that.
Yeah, that's a really good question. And I know in genetics class, my teacher is talking about bioinformatics and how technology can really expedite the process of research. One of the things she was talking about, it's somewhat irrelevant to RNA interference, but it's genetic research like the Human Genome Project and how it took them so long to do it. And now with technology, they're able to do it much faster. And hopefully one day we're going to be able to do it so that people can just walk into hospitals or like go to their doctors and have their whole genome printed out, and that would allow for more personalized medicine. But I'm really excited about the possibility and, prospect and contribution of artificial intelligence and advancements in AI and technology in general, and how that's going to impact the medical field and oncology and stuff.